Okay, we're going to talk about DNA replication. So in the last video, we introduced nucleic acids and talked about them. So nucleic acids are we're going to talk about nucleotides. We're going to talk about DNA. And they're made up of individual nucleotides that are linked together by what's called a phosphodiester bond. Sometimes you'll see this called the phosphodiester linkage. And this is the backbone of the DNA. We're talking about DNA replication. We're not talking about RNA replication. So this linkage of the nucleotides to each other is like the primary structure of the DNA. DNA is double-stranded. You could kind of think of it like a ladder where the phosphodiester backbones would be the sides of the ladder, and then there would be hydrogen-bonded purines and pyrimidines bases, which would be kind of like the rungs of the ladder. So you're going to see some pictures in the lecture, both showing DNA straight like this, like a ladder with the phosphodiester bonds, the phosphodiester linkages, like the sides, and then the hydrogen bonded bases, like the rungs of the ladder. And then you're also going to see it twisted like a double helix. So that's how it's actually exists, is twisted as a double helix. But a lot of the time, just for the simplicity of explaining DNA replication, they'll show the DNA helix straightened out. So I know that people are coming to this class from a lot of different backgrounds. Some of you have had a lot of biology and cellular and molecular biology, so this might be 100% review. Some of you have had no biology, and this is going to be all brand new. So what we're focusing on in this class is the biochemistry. We're focusing on the covalent and non-covalent interactions. We're focusing on the enzymes. We're focusing on the cellular location, that sort of thing. I have posted some videos at the links of the bottom of our Canvas page that are good kind of overviews of DNA replication. And if you are somebody that has not taken a lot of biology, I would definitely recommend starting there. And we'll talk about the specifics here. So, in Chapter 1, we introduced something that's called the central dogma of molecular biology, which shows DNA, which is the code for all of the genetic information. DNA gets what's called transcribed into RNA, specifically messenger RNA. That messenger RNA gets translated into protein. So DNA is the code, and it needs to get transcribed into messenger RNA, and then the messenger RNA gets translated into the protein. So we'll spend some time in the next few videos talking about transcription and translation. What we're talking about in this video is just DNA replication. So how and why does the DNA replicate itself? So these are processes that are easy to get confused because sometimes some of the parts are similar, but what we're talking about in this video today is replication. We'll talk about transcription and translation later. So the reason that DNA replicates itself is to make a duplicate so that when a cell divides, it has twice as much genetic information as it needs, so then when the cell separates, you still have a conserved amount of DNA. So there is a slide that shows what's called cell cycle. So I'll just draw this really quickly. And again, if you've taken a lot of biology, you know all of this, and if you haven't, this might be brand new. So we have what's called S phase. Well, let's start over here. So we'll say this is, all of this is mitosis, G1, S, G2. So G1 is when the cell grows and the DNA is duplicated, or everything else in the cell is duplicated, not the DNA. This S stands for synthesis, and it's the DNA is being duplicated. And we spend a lot of time on this in biology when we're talking about how the chromosomes separate, and all of that happens in mitosis. We talk about cell cycle regulation, how the cell knows when to go through cell cycle, when to stop, all of that. In this class, we're really talking about just how the DNA is replicating. The DNA is replicating in S phase of cell cycle. So every chromosome is replicated. So then when we get, so the cycle is going this way. So then when we get to mitosis at this point, so here we would start with one cell, and then when we would leave mitosis, we would have two identical cells to the cell that we started with. Again, this is way more complicated than this. Please, if you haven't taken a lot of biology and this is all unfamiliar, watch the videos that I've posted, kind of a crash course overview of DNA replication. So again, we care about the cellular location of all of this. DNA never leaves the nucleus. The nucleus of the cell is where the DNA is held. So DNA replication happens in the nucleus. And when we get to S phase and certain checkpoints are met and the cell knows, okay, it's time for me to double my DNA, it goes through DNA replication. And this is called semi-conservative replication. There was a lot of different hypotheses as to how DNA was replicated, and this was the way that we have discovered that DNA is replicated. So I'm going to draw the DNA like this, and then I'm going to show it kind of opening up here, because what we're going to see is this would have been like the double-stranded helix, and I've just straightened it out. So here is my straightened out DNA. And remember, DNA, the backbone has a directionality. 3', 5', and it is paired with a strand that's 5' to 3'. So new nucleotides are always added to the 3' hydroxyl. They're always added at this end. So when this opens, we're adding new nucleotides. We're using this as a template, meaning we are going to do what's called semi-conservative replication. Semi means half, which means when we get to the end of this, we have two new strands. Half of the new strand is the old, what we call parent strand. Half of the new strand is the new, what we call daughter strand. So new nucleotides can only be added to the 3' end of the growing strand. So we're gonna see this a few different ways, but let's just do it this way first. So let's say here is my code. I have A, T, C, G. So that means on the complementary side, A pairs with T, T pairs with A, C pairs with G, G pairs with C. So now it's going to start to open up. And the direction, we're going to talk about all this in more detail later, but the way these strands are replicated is different because the enzyme that adds new nucleotides, I'll just go ahead and write this now and we'll come back to it. DNA polymerase, which is the enzyme that adds new nucleotides. can only add to the 3' end of the growing chain. So if we started a new strand that was going, let's say, like this, this will be a new strand, and this will be a new strand. So what's going to happen is we're going to end up with 2 strands, 2 identical strands, and each of these new strands is going to be half parent, which is the old DNA, half daughter, which is the new DNA. The problem is the enzyme that does this, there's lots of enzymes, and we'll talk about all of that later, but before we get into that, the major thing to understand is that the DNA polymerase can only add nucleotides at the 3' end. So these strands both have a directionality because these strands are anti-parallel. So that means this is 3', this is 5', this is 5', this is 3'. So the replication is going to follow this replication fork. You can kind of think of this like a zipper, and this is going to continue to open as we go through the strand that we're trying to replicate. This is the 3' end, so DNA polymerase could continue to add in this direction because the next nucleotide that it's going to add here to the C, it's going to add a G, and at this G it would add a C on this other side. So what happens is this is the strand where the DNA polymerase is going with what we call the replication fork here. That's why this is called the leading strand. And over here, we cannot follow the replication fork because the DNA polymerase can't add here at the 5' end. It can only add here. That means this is called the lagging strand. So this is continuous synthesis. This is discontinuous synthesis. Okay, so what this looks like is the DNA polymerase would bind somewhere down here and it would just follow this replication fork as it's added. This one cannot do that. It's going to synthesize short, discontinuous pieces called Okazaki fragments. These Okazaki fragments, that's a Z, Okazaki fragments are then ligated together, but it's not following the replication fork. It's discontinuous synthesis. So this is kind of the big picture. So let's go back a little bit. So semi-conservative replication, meaning that the 2 strands of DNA that are synthesized from the 1 parent strand are half parent DNA, half daughter DNA. So there is a slide that shows some enzymes to know, and DNA polymerase is on there. Let me just go ahead and write those enzymes that you need to know. DNA polymerase, DNA helicase, topoisomerase, ligase, DNA ligase, and primase. Okay, so DNA polymerase, we already said what that does. DNA helicase separates the double-stranded helix. Topoisomerase relaxes the supercoiling. Another name for topoisomerase is gyrase. If you've ever heard of that enzyme, gyrase, these are the same 2 enzymes, they just have different names. So what happens here is when we uncoil this helix, it's kind of like if you've ever taken a jump rope that's braided together or something that's braided together and you take 2 pieces and you pull it apart. They're super coiling on either sides of what you've separated. So what the topoisomerase does is it relaxes that supercoiling by making little cuts and then putting it back together so we don't have all kinds of supercoiling on either side of where the replication is happening. Ligase is reannealing these Okazaki fragments. And then primase provides a starting point of RNA for DNA polymerase. So another odd thing is that DNA polymerase cannot initiate DNA transcription. DNA polymerase has to have an RNA primer, and that RNA primer is then come back, later the enzymes will come back and remove those RNA bases that were initially started. But when we're starting DNA replication, we have to start with a primer which is made of RNA nucleotides. So there's a lot of good pictures in here of exactly what is happening. The thing that is most important for us to recognize is the directionality of this process. And the reason that this is important, the reason that we can only add new nucleotides to the 3' direction is because the DNA polymerase needs the free hydroxyl on the 3' carbon. So this is where the polymerase reaction happens, where the condensation reaction happens. This hydroxyl group is going to react with the incoming nucleotide to form that phosphodiester bond. And that happens on the 3' carbon. The 5' carbon does not have an available site for this reaction to happen. At the 5' carbon is that big phosphate group from the previous phosphate, the phosphodiester bond. So this is why nucleotides can only be added at the 3' end of the growing strand. So we want to know where it's happening, why it's happening, kind of the steps to the process of this, and the reason that there is a directionality to this process is because the DNA polymerase will only add at the 3' carbon because it needs that free hydroxyl group for the new bases to be added to. Again, watch those videos if you've not taken a lot of biology prior to this, and we'll continue talking about DNA when we talk about transcription and translation.